Hello viewers. In today's lecture, I am going to discuss about the topic endocrine pancreas. Before I start, I would like to mention that this video is the part of the initiative taken up by the Zoological Society of Assam to help the students pursuing their graduation in zoology. I want to dedicate this video lecture to my beloved parents who are my constant support. So, let's start our lecture. The learning outcomes or objectives. At the end of this lecture session, the students will acquire knowledge on the following. They will get to know about the pancreas, its functions as both exocrine gland, thereby releasing its digestive enzymes through its ducts, and endocrine gland by secreting various hormones. They will get to know about the structure and position of pancreas. They will also get to know about the histology of pancreas. The students will get to know about a brief overview of exocrine pancreas, and finally the students will know about the structure, composition and function of endocrine pancreas that consist of various cells, that is alpha cells, beta cells, delta cells, pp cells and epsilon cells. So, following these objectives, let us begin our today's discussion. Let us have a look on the historical background. The discovery of pancreas is attributed to a Greek anatomist and surgeon Herophilus of Chalcedon. In 19th century, the role of pancreas in digestion was established by a French physiologist, Claude Bernard. He mentioned about the function of the pancreas in digestive processes. Johann Georg Wirsung from University of Padua discovered the pancreatic duct. He also stated that the pancreatic duct joins or perforates the duodenum. In 1869, Paul Langerhans, a student of medicine, discovered small polygonal cells that appeared in small clusters in the parenchyma of pancreas. Later, they were named as islets of Langerhans. Again, in the year 1921, Banting and Best discovered insulin at University of Toronto. Frederick Grant Banting and John James Ricard MacLeod in 1923 received Nobel Prize in Physiology or Medicine for the discovery of insulin. Banting approached MacLeod to conduct studies on insulin, and MacLeod finally accepted his proposal. Best was a medical student and he helped Banting in the discovery of insulin. Polyp, who joined the team, developed a method that helped in the extraction of purified insulin in large quantity. Banting, Best and Polyp were also granted patent rights for the production of insulin. Kimball and Moorlin in the year 1923 reported a hyperglycemic hormone which was later named as glucagon, and its molecular structure was established in late 1950s. Next comes the pancreas. Pancreas is a heterocrine gland, that is, it is a mixed gland and functions as both exocrine and endocrine gland. The exocrine gland comprises of acinar cells that performs the digestive functions. Exocrine region forms the bulk region of the pancreas, that is 80% of the pancreas. Endocrine pancreas occupies 2% of the pancreatic tissue. The endocrine pancreas comprises of islets of Langerhans that secrete different kinds of hormones. They are insulin, glucagon, amylin, somatostatin, pancreatic polypeptide, and ghrelin. Islets of pancreas plays an important role in glucose homeostasis. Therefore, it helps in maintaining the blood glucose level. Next comes structure and position of pancreas. The position of the pancreas is parallel to and below the stomach. In this pictorial diagram, you can see the pancreas and its position. This is the duodenum, this is the pancreas, this is the spleen, this is the pancreatic duct, this is the common bile duct, this is the common hepatic duct, this is the right and left hepatic duct of liver, this is the gallbladder, this is the cystic duct, and this is the accessory pancreatic duct, and this is the jejunum. Pancreas is divided mainly into 4 portions: head, neck, body, and tail. The head lies in the loop of the duodenum. You can see in the picture here. The body lies posteriorly to the distal portion of the stomach, and the tail stretches up to the spleen. The pancreas basically consists of pancreatic acini, islets of Langerhans, and various ducts. We shall discuss subsequently it in the coming slides. The pancreatic enzymes flows through the main pancreatic duct that connects the hepatic duct before unloading its various enzymes into the duodenum through the papilla of Vater. Papilla of Vater is the point where the junction of pancreatic and bile duct enters into the duodenum, that is surrounded by sphincter of Oddi, which controls the flow of the digestive juices. Next comes histology of pancreas. The pancreas is enclosed by a capsule made up of loose connective tissue. Again, pancreas is divided into distinctive lobules. The pancreatic ducts are connected to each other for the flow of pancreatic enzymes successively through intercalated ducts, intralobular ducts, interlobular ducts, and main pancreatic duct that finally reaches the duodenum. So in the figure, you can see the intercalated duct, interlobular duct, intralobular duct that finally connects the main pancreatic duct for the flow of enzymes. The pancreas is richly supplied with arteries and veins. In the figure, you can see the presence of blood vessel. Pancreatic acini are present in clusters. You can see in the figure the presence of pancreatic acini. These are the pancreatic acini. They are pyramidal in shape and consist of zymogen granules that contains proenzymes. Let me say what proenzymes are. When enzymes are present in inactivated form, they are known as proenzymes. The islets of Langerhans are usually small. This is a islet of Langerhans. Let us now have a look on a brief overview of exocrine pancreas. The exocrine pancreas produces digestive enzymes or pancreatic juices secreted from pancreatic acini in response to the existence of chyme in small intestine. You may wonder, what is chyme? Chyme is an acidic fluid that is passed from the stomach to small intestine. Pancreatic juice mainly contains water, enzymes, and bicarbonate ions. The bicarbonate ions helps to neutralize the acidity of chyme released from the stomach. It is to be noted that the bicarbonate ions are secreted from the ducts of the pancreas. The pancreatic secretions are mainly controlled by nervous system and hormones. Protein digesting pancreatic enzymes are trypsin, chymotrypsin, and carboxypolypeptidase. Carbohydrate digesting pancreatic enzyme is pancreatic amylase. Fat digesting pancreatic enzymes are pancreatic lipase, cholesterol esterase, and phospholipase. Next comes the endocrine pancreas. The endocrine pancreas consists of islets of Langerhans that comprises of various cells. Beta cells constituting 60% of the islet and secrete insulin and amylin. Alpha cells constituting 25% of the islet and secrete glucagon. Delta cells constituting 10% of the islet and secrete somatostatin. Pancreatic polypeptide cells, also known as PP cells, are present in less amount and secrete pancreatic polypeptide. Epsilon cells are also present in less quantity and secrete ghrelin. Insulin mainly controls carbohydrate, protein, and fat metabolism. Improper functioning of insulin causes acidosis, that is, presence of acid in blood, and arteriosclerosis. In arteriosclerosis, the walls of the arteries become thick and hard. Improper functioning of insulin also causes wasting of tissues and causes cellular deformities and even causes death. The cells of the islets are present close to one another and interact among themselves for cell-to-cell communication and controls the secretion of hormones. Next comes structure of islets of Langerhans. Islets of Langerhans mainly comprises of alpha cells, beta cells, delta cells, PP cells, and epsilon cells. Most of the islets vary in size between 100 to 150 micrometer in case of human in diameter. Larger islets are distributed beside the main pancreatic duct and interlobular duct, whereas the smaller islets are found scattered among the acinar cells of exocrine pancreas. Islets are spherical, ellipsoid, and are irregular in shape. Islets are mostly found in the tail of the pancreas. Islets may be also found in the other portions of the pancreas too. In the figure, we can see a section of pancreas showing islets of Langerhans. Here, the pancreas section is stained with Gomory's chrome alarm hematoxylin floxin stain. This stain is mainly used to differentiate alpha cells secreting glucagon and beta cells secreting insulin. Alpha cells are stained pink and beta cells are stained blue. Islets number count in an adult human is 1 to 2 million. Beta cells are found in the center of the islets and alpha cells surrounds the beta cells, and PP cells are present on the periphery of the islets. Islets are abundantly supplied with blood capillaries so as to secrete various hormones directly into the bloodstream. Now, let me discuss about the different types of cells of islets of Langerhans. Beta cells. Staining of beta cells are particularly done with aldehyde fusin and pseudoisocyanine, followed by oxidation. Immunofluorescent techniques establish that insulin is produced and is secreted from the beta cells of pancreas. Beta cells are small, numerous, carry different secretory granules with dark center and has angular insulin granules. Next comes alpha cells. They are normally acidophilic, which means they stain with acid dyes, and argyrophilic, which means they stain by silver salts. They do not stain by the method of aldehyde fusin and pseudoisocyanine. Alpha cells secrete other peptide hormones along with glucagon in some species. Alpha cells contains round secretory granules. They are generally larger than beta cells, and the granules are uniform in size, consisting of large dark center. Then comes delta cells. They are elongated, and another important point is that they possess filopodia-like extension. It can be distinguished from alpha cells by toluidine blue staining procedure. They are further differentiated from alpha cells and beta cells by staining with pseudoisocyanine following methylation. Therefore, this allows clear visibility of delta cells. They have large secretory granules than alpha cells and beta cells. Then comes PP cells. They are generally localized in the islet periphery and also in the cells that is distributed all over the exocrine pancreas. They can be distinguished from the other cell types cytologically as well as immunologically. Then comes epsilon cells. They are located in the pancreatic islets and are present in very negligible amount. Now, in this figure, we can see the various types of cells of islet of Langerhans. These are beta cells. You can see in the figure here, they are green in color and are numerous. Alpha cells lie towards the periphery. Delta cells are elongated in shape. Apps cells, also known as PP cells that secrete pancreatic polypeptide, also lie in the periphery of the islet. Here is the exocrine pancreas consisting of acinar cells and duct cells. Again, in this figure, we can see the secretion of hormones from islet of Langerhans of the pancreas. Hormones are secreted into the blood and are transported to their respective targets. Next comes structure and functions of the hormones of endocrine pancreas. Starting with insulin, let me talk about its structure. Insulin is secreted and synthesized from the beta cells of pancreas. Let me discuss about synthesis of insulin. Pre-proinsulin is formed by the translation of insulin RNA by the ribosomes attached to endoplasmic reticulum. Then pre-proinsulin is cleaved in the endoplasmic reticulum to form proinsulin that consists of three peptide chains, that is A, B and C. Proinsulin is then further cleaved in the Golgi apparatus to form insulin having A and B chain. Insulin is a small protein hormone and has a molecular weight of 5808 daltons, that is in case of human insulin. Insulin consists of two chains made of amino acids, that is A chain that has 21 residue and B chain having 30 residue connected to each other by disulfide linkage and C peptide. You can see in the figure the structure of insulin. This is A chain, this is the B chain and this is the C peptide. Coming to the functions of insulin. Insulin is anabolic. It helps in the quick uptake and storage of glucose, fats and amino acids in the cells and tissues, thus lowering blood glucose level, creating a hypoglycemic condition. Insulin helps in the transport of glucose into the muscle cells and is stored in the form of muscle glycogen if the muscles are not exercised after a meal. Insulin helps in the uptake of glucose by liver and is stored as glycogen in the liver. Insulin has a role in carbohydrate metabolism. It helps in storing extra energy, that is excess amount of carbohydrate is stored in the form of glycogen in the liver cells and muscle cells. Surplus amount of glycogen are converted into fats by insulin and is stored in adipose tissue. Decrease in blood glucose concentration also decreases the secretion of insulin and increase the secretion of glucagon hormone, which splits glycogen into glucose, and thus the glucose diffuses back into the blood. Again, insulin affects fat metabolism. Lack of insulin can cause heart attacks, cerebral stroke and vascular accidents. It converts excess glycogen into fatty acids to be stored in the adipose tissue. In other words, when glucose quantity is more and cannot be stored in the form of glycogen, insulin then converts excess glucose to fatty acids. Further, these fatty acids are packed as triglycerides in very low density lipoproteins and transported in this manner in blood to adipose tissue and are stored as fat. Insulin also forbids gluconeogenesis in liver. I would like to mention that gluconeogenesis is a metabolic pathway that generates glucose from non-carbohydrate precursor. Insufficient insulin increases the level of plasma cholesterol and phospholipids. Diabetic people with high cholesterol level can develop atherosclerosis. Insulin affects protein metabolism. Insulin helps in the transportation of amino acids into the cells and prevents protein catabolism. Insulin, along with growth hormone, helps to promote growth. It was found in an experiment that administration of either growth hormone or insulin at one time did not promote growth. Thus, insulin along with growth hormone work synergistically to promote growth. Absence of insulin causes ketosis, meaning formation of ketone bodies in body fluids in large quantities, and acidosis, that is production of acetoacetic acid and beta hydroxybutyric acid in body fluids. It is especially seen during severe diabetic conditions. Insulin prevents lipemia. In lipemia, there is a rise in plasma fatty acid concentration. Let me now talk about mechanism of secretion of insulin. Rise in blood glucose concentration controls the secretion of insulin. Whenever there is a hyperglycemic condition, which means increase in glucose concentration in blood, insulin gets secreted out from the beta cells of pancreas. Here in this figure, you can see that glucose enters the cell through GLUT2, which is a glucose transporter present in beta cells of pancreatic islet. There are many glucose transporters which are present in different cells and organs. Glucose is then phosphorylated by glucokinase and is metabolized to pyruvate. Pyruvate enters the mitochondria and then enters citric acid cycle to get metabolized. ATP so formed in oxidative phosphorylation inhibits ATP-sensitive potassium channels. Depolarization of beta cells increases calcium ion influx. You can see calcium ions entering the cell. Insulin is released by exocytosis due to the stimulation made by calcium ions. Glutamate is also formed. It helps to prime secretory granules and prepares the secretory granules for exocytosis. Next comes factors and conditions that regulate insulin secretion. Insulin secretion is increased by increased blood glucose, increased blood-free fatty acids, increased blood amino acids, gastrointestinal hormones, glucagon, growth hormone, cortisol, parasympathetic stimulation, acetylcholine, beta-adrenergic stimulation, insulin resistance, obesity, sulfonylurea drugs. Insulin secretion is decreased by decreased blood glucose, fasting, somatostatin, alpha-adrenergic activity, and leptin. Next is glucagon. Let us see its structure. Glucagon is catabolic and is secreted from the alpha cells of pancreas. You can see in the figure the structure of glucagon. It is a large polypeptide hormone consisting of 29 amino acids and is made up of a single chain. It has a molecular weight of 3485 daltons. It is secreted during hypoglycemic condition. That means when the blood glucose level is low and is called as hyperglycemic hormone. The structures of all mammalian glucagon are similar. Next comes functions of glucagon. Glucagon increases blood glucose level. Glucagon helps in glycogenolysis, that is breakdown of glycogen into glucose, which is the simple form of sugar that helps in the production of energy. Another important point is that glucagon helps to increase gluconeogenesis. Glucagon facilitates the activation of adipose cell lipase. So, fatty acids becomes available to the energy system. Glucagon inhibits the storage of triglycerides in liver. As a result, the liver is prevented to remove fatty acids into the blood. Glucagon upgrades the strength of the heart. Glucagon increases blood flow in kidney. Glucagon also helps to intensify bile secretion. Glucagon inhibits the secretion of gastric acid. Glucagon also stimulates lipolysis in liver and adipose tissue. Next comes regulation of secretion of glucagon. The stimulators of glucagon secretion are amino acids, cholecystokinin, gastrin, cortisol, exercise, infections, other stresses, beta-adrenergic stimulators, theophylline, acetylcholine. The inhibitors of glucagon secretion are glucose, somatostatin, secretin, FFA meaning free fatty acid, ketones, insulin, phenytoin, alpha adrenergic stimulators and GABA. Next comes somatostatin. Let me talk about its structure. Somatostatin is secreted from the delta cells of the pancreas. Somatostatin has two forms, somatostatin 14 and somatostatin 28. In the figure, you can see the two forms of somatostatin, that is somatostatin 14 and somatostatin 28. Somatostatin 28 is found to be more active than somatostatin 14. Next comes functions of somatostatin. Somatostatin inhibits the secretion of insulin, glucagon and pancreatic polypeptide. Somatostatin acts locally in the pancreatic islets in a paracrine manner, thereby acting on the nearby cells. Somatostatin also helps to decrease the motility of stomach, duodenum and gallbladder. Somatostatin also decreases the secretion and absorption of GI tract. Factors that stimulates the secretion of somatostatin are rise in blood glucose, increased level of amino acids, increase in amount of fatty acids, increased concentration of many gastrointestinal hormones that are released from the upper region of GI tract in response to ingestion of food. Next comes amylin. Let me talk about its structure. Amylin is a polypeptide hormone having 37 residue of amino acids. It is another hormone that is secreted from the beta cells of pancreas and also known as islet amyloid polypeptide, that is IAPP. In the figure, you can see the structure of amylin. Next comes functions of amylin. It is fascinating to know that amylin has an effect on glucose metabolism. Amylin helps to normalize the glycogen content in liver during diabetes. Amylin also inhibits glucagon secretion. Amylin delays gastric emptying, meaning the contents of the stomach are moved slowly into the duodenum. Amylin also inhibits food intake and decreases body weight. Amylin acts as a means in providing satiety. Factors that stimulate the secretion of amylin. Nutrient stimuli generally causes secretion of amylin, which circulates in glycosylated as well as in non-glycosylated form. The distribution of amylin is similar to that of insulin in plasma as well as in interstitial fluids. Next comes pancreatic polypeptide. Let me discuss about its structure. Pancreatic polypeptide is a linear polypeptide consisting of 36 amino acids. It has a molecular weight of 4200 daltons. It is produced from PP cells of the pancreatic islet. They are found mostly within the head of the pancreas. In the figure, you can see the structure of pancreatic polypeptide. Next comes functions of pancreatic polypeptide. Pancreatic polypeptide inhibits pancreatic secretions, bicarbonates, and enzymes. Pancreatic polypeptide helps to stimulate gastric juice secretion. It is antagonist to cholecystokinin. Cholecystokinin is secreted from duodenum that stimulates the release of bile and secretion of enzymes from the pancreas. It also slows down the absorption of food in humans. Factors that stimulates the secretion of pancreatic polypeptide are intake of protein meal, fasting, exercise, and acute hypoglycemia increases the secretion of pancreatic polypeptide. Its level is decreased by somatostatin and intravenous glucose. Next comes ghrelin. Let me talk about its structure. Ghrelin is secreted from the epsilon cells. It is a polypeptide made of 28 amino acids. The cells of ghrelin are ovoid in shape. In the figure, you can see the structure of ghrelin consisting of 28 amino acids. Next comes functions of ghrelin. Ghrelin stimulates the secretion of growth hormone. Administration of ghrelin induces food intake by increasing the appetite. Ghrelin maintains energy homeostasis by regulating body weight. Ghrelin administrates gastric secretion and gastrointestinal motility. It plays an important role in glucose homeostasis and modulates pancreatic secretions. Ghrelin diverges cardiovascular functions by decreasing arterial pressure without changing the heart rate. It also exerts anti-inflammatory functions by inhibition of pro-inflammatory cytokines. Ghrelin controls reproductive functions. That is, ghrelin controls reproductive physiology as well as pathology by endocrine and paracrine pathways in both male and female. Ghrelin induces bone formation by stimulating the osteoblastic cell proliferation and differentiation, including inhibition of cell apoptosis and increasing the minerals. Factors that stimulates the secretion of ghrelin are hypothalamic neurons present in arcuate nucleus. The region that controls food intake stimulates the expression of ghrelin. Next comes commonly occurring disorders of pancreas. Acute pancreatitis. It is a severe condition where the ducts of the pancreas are blocked. The pancreatic secretions are then pulled in the areas that gets damaged. In such a situation, the pancreatic secretions becomes activated and can digest the whole pancreas. It may be sometimes lethal. Chronic pancreatitis. It is a progressive disorder that damages the pancreas, which finally leads to diabetes. Hereditary pancreatitis. It occurs due to genetic mutation that can cause chronic pancreatitis. Diabetes mellitus. It is a global health issue. It is an endocrine disorder that can cause dysfunction of carbohydrate, fat, and protein metabolism. They are generally of two types. Type 1 diabetes. It is also known as insulin-dependent diabetes mellitus, and it occurs due to lack of insulin secretion. Here, the beta cells of the pancreas are destroyed by viral infections or autoimmune disorders. It is usually referred as juvenile diabetes due to its onset in early period of life, but it can occur at any age. It raises the blood glucose level and increases the amount of cholesterol and causes retinopathy, kidney diseases, stroke, heart attack, etc. Another type of diabetes mellitus is type 2 diabetes. It is also known as non-insulin dependent diabetes mellitus that occurs due to resistance of insulin in the target organs. It occurs commonly among 90 to 95% of people in adult stage and is also called as adult onset diabetes. Weight gain and obesity can risk the development of type 2 diabetes and also causes lipid abnormalities. Metabolic stress also enhances the aging of beta cells. Insulinoma, hyperinsulinism. Increased insulin production occurs due to adenoma, that is, tumor formation in the islets of Langerhans, of which 10 to 15% of adenoma are malignant. Thus, metastasis from the islets mostly spreads throughout the body, causing excessive insulin production, and thus hypoglycemic condition occurs in the body. Insulin shock. In this case, high level of insulin in blood is produced from insulin secreting tumors or by administration of insulin in the body, and the blood glucose level falls that causes central nervous system to excite, resulting hallucination and nervousness, and thus it may even cause coma. Exocrine pancreatic insufficiency. In this case, pancreatic enzymes becomes deficient and causes weight loss, bloating and others. Cystic fibrosis. It is due to genetical cause that affects the lungs, digestive tract and pancreas. These are the suggested readings and references that I have followed for the preparation of this video lecture, and you may go through it for more information. Some references I have mentioned here also. So with this we have come to the end of this video lecture. For any queries and feedback, contact me at my email address rajashreedeka41@gmail.com. I would sincerely acknowledge and be thankful to Zoological Society of Assam for giving me this platform. Thank you so much for your kind attention and wish you all the best.